Assalamualaikum, we are from Group 9 and today we are going to present molecular techniques on conserving biodiversity. This presentation focuses on past effort of conserving biodiversity in Malaysia. So let's get started. First, let's meet our group mates. First, Nurfarahin, which is me, MARDHIAH, AFIFAH ILIYANA, AMEERA FATIN FARHANA, and SYAHIRA.
The future biodiversity and its significance to Earth and human are becoming a concern. This triggers the development of importance of molecular techniques from time to time. This is due to issues such as genetic issues that give impact to conservation of biodiversity. Skills can be developed via modern molecular techniques, including cloning, in vitro propagation, cryopreservation in ex situ programs, polymerase chain reaction, DNA sequencing, and many more. Hence, those that treated with excision can be helped to defeat several abiotic or biotic risks. This is crucial to define the relationships among individuals, species, and populations in conserving biodiversity.
This molecular technique is adapted from journal written by Joseph et al. In 2019. Due to the massive turtle slaughter at Pulau Tiga, Malaysia, the researcher do a molecular technique to estimate the origin of sea turtle carcasses found at Pulau Tiga, Kudat, Malaysia in 2014. This study aims to determine the species composition and size frequency of sea turtle carcasses found on the Pulau Tiga island. Also, using genetic technique, carcasses are assigned firstly to rock crease, which is to determine their natal origin, and second is to foraging ground, which is to estimate where poaching occurred. The result provide insight regarding the sea turtles migration and also the potential effect of poaching on sea turtle occurred. The molecular technique applied in this research is two-step polymerase chain reaction (PCR) protocols. Genomic DNA was extracted using the CTAB protocol. First, a segment of the mitochondrial control region of approximately 800 pp was amplified from the extracted DNA using primers LCM 15382 and H950G. Using the first PCR product as template, it was re-amplified by semi-nested PCR ususing primer LTEI9 and H950G. The PCR amplification was performed using an append-off master cycler DNA engine thermocycler PCR. Then, all amplified samples were verified for the targeted band size by 1% agarose-gel electrophoresis. Finally, the PCR product was sent to the first base, Kuala Lumpur, Malaysia, for purification and sequencing of both strands. The sequences were truncated to approximately 770 base per region that has been widely evaluated in the recent study of sea turtle. Haplotype were identified by performing a search using the program BLAST in the NCBI, National Centre for Biotechnology Information. The Southwest Fisheries Science Centre, NOAA Fisheries Service, was also consulted for anti-DNA sequences of green turtle from the Pacific and the Indian Ocean. This information not only support the MSA estimation of natal origin, but also provide direct evidence of a mortality factor for nesting green turtle at the turtle island. Overexploitation is one of the numerous threats for sea turtle conservation, which include poaching of eggs and meat, incidental captures, entanglement, and also boat collision. Identifying the location of poaching and its effect is becoming important for sea turtle conservation and law enforcement. This study applied molecular technique that have been prevalent not only in ecological study but also in wildlife forensic. Similar technique in combination with the development of genetic marker for more precise geographic resolution such as mitochondrial short tandem repeats will further contribute to the development of measures against poaching activities.
This biomolecular technique, according to research by Baston Gokon, the project focused on stem cell technology and in vitro fertilization. The process is similar to cloning technology in that it aims to give birth to a new baby using cells from old rhinos. Iman, the last Sumatran rhino, died last November, bringing the endangered species one step closer to extinction. Now, scientists are hoping to use tissues and cells from Iman and other dead rhinos to bring the population back. The scientists collected cells and tissues from the last 3 Sumatran rhinos at Bora Sanctuary, including Iman, before each died. The cells came from the rhino's hearts, lungs, brains, and kidneys. The team collected stem cells, basically raw material from which cells with specialized functions can be generated. The first is to develop these stem cells into an egg and sperms to create an embryo that will be implanted into a surrogate mother. The surrogate will likely be another rhino, either a Sumatran rhino from another country or another species. The second method is to take the egg of a surrogate animal, remove the nucleus, and join it with a Sumatran rhino somatic cell. They need to analyze the cells to create a genomic database, differentiate the stem cells, and work with zoos to find a suitable surrogate female. Last year, scientists successfully fertilized in vitro embryos collected from the 2 remaining females with sperm from dead males. The conservation efforts are to establish a captive breeding stock nearly extinguished the species. Sumatran rhino rescue effort, a few young adults have been born in captivity. A protection conservation program is designed to protect rhinos from poaching and hunting. The program will work to increase security for rhinos, and it will also aim to raise awareness among the community about the importance of protecting these animals. These are critically endangered due to hunting for their horns and the destruction of their habitat.
Not only animals, Malaysia also make conservation effort to conserve plants and trees. Belian wood is the most famous wood due to its durability, and it is labeled as endangered in Sarawak. In Malaysia, there are found two species, which is E. zwagari and E. malanggangai. So, before the conservation effort needs to be done, it is important to distinguish which species is endangered, and recent and other researchers studies on this species. Their objectives are to genotype the Belian wood using M30 universal primer and even E. zwagari scar marker. Overall genotyping procedure are listed below. First, the samples were collected from Lendu and Sarawak. Second step is total genomic DNA of both species are isolated with CTEP method. The third step is the DNA concentration determined using agarose gel electrophoresis. The fourth step is the DNA amplification, which is the most important part. And last but not least, the PCR product are separated on agarose gel electrophoresis before the band are being observed. To get depth into the PCR amplification, the PCR component they use are genomic DNA, PCR buffer, magnesium chloride, DNTP, M30 universal primer, and tag DNA polymerase. M30 primers are used to insert clone into the M30 vectors and PUC plasmid developed by Messing. The amplification were done in thermocycler for 35 cycles of 1 minute denaturing, 1 minute of annealing, 2 minutes of extension, and final extension for 10 minutes. So, what is scar markers? The sequence characterized amplified region or scar markers are developed to produce the specific and reproducible results that could be used for species or cultivar identification. To achieve the second objective, the diagnostic fragment were identified from the DNA profile are extracted from the agarose gel electrophoresis before purified. Next, the fragments are cloned into the vector before undergoes blue-white screening for identification of recombinant DNA. The next step is extraction of plasmid before sequencing. Lastly, the scar marker of E. zwagari was developed.
Molecular techniques are indispensable and are increasingly being used to address questions of conservation relevance. Molecular biology is undoubtedly the fastest evolving field of science. Some importance and challenges in molecular technique on conserving biodiversity is that they are particularly useful for studying rare species whose cryptic habits make them challenging study subjects when using traditional field methods. In fragmented environments, distributions and population connections can be analyzed using molecular methods to pinpoint populations that require immediate conservation attention. Thirdly, by enabling conservation biologists to swiftly scan a wide variety of individual and population traits at a given site, molecular biology is aiding in the reversal of that tendency. Some challenges in this case are how many different classes are present, and estimation of genetic could be tackled using molecular techniques. Secondly, there is a trade-off between different types of markers with regard to their use for diversity assessments. Lastly, is less facilities for microsatellite cloning and sequencing to enable researchers with access to the best data. That's all from us. Thank you for your attention.